Rick? Oh, there it is. Okay, I think we're live. That's awesome. So, my name is Katz, and this is the first episode of PodCatZ. And I am joined with Dr. Allan Drummond.
Drummond.
Drummond. Yes. Wonderful. And I feel like it's going to be so difficult to accurately represent what you do, but I'll try reading from here. He's an associate professor of biochemistry and molecular biology and genetic medicine at University of Chicago. But a lot more than that too, right? As awesome as that already sounds, you're also an artist and you did some other work. Why don't you tell me about like the stuff that you've done to get to where you are?
The sort of condensed life story. I guess. So I grew up mostly in Iowa. Mm-hmm. Graduated high school in Iowa. Went to school out at Princeton, and got a degree in mechanical engineering. And then didn't want to be a mechanical engineer, so I chose between two startup companies, like a 3D games company and a e-commerce software company. Chose the e-commerce software company as a startup in 1995. This was like back in the day when Amazon sold books and people wondered whether you would buy things on the internet. So I'm old. Spent 7 years there. Ended up being a human resources director,
cool.
Of all things. And yet would go home at night and read textbooks on quantitative biology and complexity theory and quantum mechanics and all this other stuff. I'm sure you know the kind of the headspace that one can get into. And decided, my HR advice to myself is you're in the wrong job. So
that's so cool.
I applied to graduate schools. Longer story that we can maybe get into, but they all rejected me, and I was.
But hold up, why were you rejected? Just because no prior studies in the field, or?
Well, I mean, there's the sort of details of the story, which is that I was actually invited to interview at Caltech, and got into the first interview, and Shuki Brook, one of the professors, asked me a typical brain teaser. He said, that if A is greater than B and B is greater than C, that A is greater than C, of course, right? That's the transitive property. I said, yes, I know this. He says, okay, now imagine they're random variables. And I tell you that the probability that A is greater than B is greater than 50%, and the probability that B is greater than C is greater than 50%. What can you now tell me about the probability that A is greater than C? And I said, I said a bunch of stuff, right? But it really didn't make much sense. Anyway, so I had a bunch of other interviews with people.
Wait, what was the answer? Don't. Yeah, what's? We'll go back.
Think about it for a while, but let me say it's just it's an interesting problem, and it's not, and the answer is not what most people jump to immediately. Anyway, the rest of the people who interviewed me, the other professors, asked me a bunch of kind of softball questions about my interests and so on. And I don't know, I'm articulate, right? So they had a great time. Then behind the scenes, when they were looking to discuss me, everybody said, this guy seems great. And then Shuki said, well, I mean, this is what I heard. a little bird told me, right, like along the chain, that he comes from this really weird background. He doesn't have any research experience. He's worked for this software company. He's a human resources guy. He's utterly different on paper than anybody that was in the engineering class. He said, did anybody else ask him anything technical to assess his technical capability? And everybody was like, no. He said, well, I did, and he's terrible. So I got told this story by a little bird, and I went, oh, that's really depressing, because of course I feel like I have the technical capability to do this, but I understand in the context of an interview, that's not the way it goes. So I was, so they rejected me, right? Net was 80% of people who get an interview on campus typically will get invited to join the graduate program, and I was in the unlucky 20%. And so I was completely devastated by this. Then went back to campus, or actually just reached out and said, look, I understand the decision. I'm incredibly passionate about this, because my emotional reaction to the rejection was not like, oh, it's very disappointing that I was being rejected. It's like this entire life path has been cut off for me. And I realized at that point that was really not an option. Like, I needed to do something in this space. I needed to become a scientist. And so I said, I understand the decision, don't want to change the decision, but what can I do in order to improve my competitiveness for the next time around? One conversation led to another, and eventually they said, well, let's just give you another round of interviews. So they gave me another round of interviews, reversed their decision, and then admitted me that same year.
Nice. That's awesome.
there, then, fast forward 4 years later, I graduated with a PhD, won the thesis prize.
But what was the doctor that originally rejected then?
What was the
name? Yeah, who?
Oh, Shuki Brook. He's great. I mean,
Shuki Brook. So you then had to like hang out with Shuki Brook and do work with Shuki Brook. Was it awkward?
Like I said, no.
Not at all.
Not at all. No.
Okay.
I understand. I'd actually, I'd worked for this company Trilogy for 7 years and done hardcore recruiting of incredibly smart kids for a long time, and I had given them the exact same brain teasers, right? And the subjective, so I knew the game, right? Like it's fine. It was nothing personal for him, and he turns out, first of all, he's bloody brilliant. Second of all, he's super warm and very thoughtful about developing his students and thinking about this. He's great. So it was fine. No harm, no foul.
Okay, cool. And from his end, then everything was cool. He never reached out and was like, hey, like.
No.
Okay.
No. I mean, I think he's smile and nod and pat me on the head and then hope that I do well, but it wasn't clear that I was going to. But then I did, right? So I graduated, got my PhD, and was fortunate enough to win an independent fellowship at Harvard to start a lab there.
How does that work? How did you win that?
Yeah, so obviously it's competitive.
Yeah, when you say Harvard and
Yeah, so I mentioned in passing. So my thesis work went very well. I published 5 papers as a graduate student, and several of them made a significant impact, I think, in the field. And so I'd come to the attention of people. I was a theorist at the time, so just doing paper and pencil, and computational type of work. And Harvard is looking for people doing systems biology, right? This is the Center for Systems Biology, and they're particularly, I think, attracted to people who were making a transition from, say, theory to experiment or vice versa, where their particular, surroundings and mentorship available there was going to make a big difference in those people's careers. And that was exactly what I was proposing. I said that, I'm a theorist. I've proposed these hypotheses. They're testable. They're empirically testable. You can do experiments that are going to get to the bottom of this, but no one's going to do them except for me. So, and I want to throw myself, wholly into this, set up a lab and start doing experiments, which is as you can probably guess, making the gamer caster kind of thing, right? They're related in some way, but they're utterly different skill sets.
Of course.
Right?
So this is the start of the Drummond Lab?
That's right. Yep.
Awesome. And it's still standing today. Is it
That's
still
right.
a Harvard endeavor then? Or are they still...
So the Harvard position was 5 years, and then you're out, right? That's
cool.
The idea. It's not a tenure track.
Cool. But then what did they facilitate then? Was it like...
I got to set up my lab. And so it's not really, you do have a physical lab, of course, but it's more like, hire people, get a graduate students, right? So my first graduate student who joined me in that first year, she's now an assistant professor at Arizona State University doing evolution. Sort of turned out great.
Awesome.
So you build a lab. It's like you built a team, right? So it's that kind of thing, right? Let's select the people and try to then do your thing, right? Try to succeed. And
so
then,
Harvard was like you would pull from human resources from Harvard.
It's the typical. No, it's much more informal than that, right? So they're like my first technician, who was really transformative, because I couldn't do anything experimentally. I'm terrible, but I'm super interested in it, and I can evaluate if it's gone well or not. It's sort of like being an art critic versus an artist, right? So I got him because his previous mentor was leaving actually to go to the University of Massachusetts, and he didn't want to leave Boston.
Cool.
And so I said, well, join my lab. And he just immediately was this powerhouse in the group, able to do things that, if I could imagine it, he could figure out some way to do it.
That's so cool.
Awesome.
Practical people are incredible.
Yep.
So, I'm going to pull up the website here for the Drummond Lab. And when I clicked on your Twitter profile, and I've clicked on it a few times, right? But I clicked on the website and I started reading this, and I'm going to read it out loud for a little bit, if that's okay. It says, we study how cells adapt at the molecular level, focusing on the process of translation of cellular genetic information into protein molecules. Our analysis extends from the near instantaneous cellular responses to environmental stress to the multi-million year evolutionary divergence of species. Which seems like a really cool, well-packed summary. I was telling you this the other the other day, and it's just like, you read that and you're like, whoa, that sounds awesome. Obviously, I can't quite wrap myself around everything that means, and even as far as like making it readable and legible for someone that's maybe foreign to this type of research or lab. It sounds like something that I can sort of grasp, but not like each word is like, whoa, okay, like that, that sounds cool, and that sounds cool, and that sounds cool. But how does it all come together?
And the string of them together.
Yeah, exactly.
Yeah. No, that's great. I'm glad that it lands that way. And, every word is true in there. And I think there is this, part of building a lab is building a kind of description of yourself and the sort of ambitions that you have and the vision that you have that is going to attract the right kind of talent, right, to do the things that you want to do. And we are very much we're hardcore biochemists at one level, but in service of, we're very conceptual, right? Like we like to think big, we like to challenge the way people think about their individual biological problems, and we like to use whatever tool is required to get the point across. And so that paragraph is trying to capture that.
That's pretty awesome.
For you, right?
I love it. I'd love to talk about this. I want this conversation to be as free-flowing as possible. There is no time restraint on my end, so please feel free to talk about whatever you want, whenever you want.
Absolutely in your hands. I just want to say, just to finish the story, to bring people up to the present day, right? Like moved the lab from Harvard to the University of Chicago in 2011, and that's starting a typical assistant professor position on the tenure track, and got tenure a couple of years ago. And so I'm now in the sort of career stage of being a junior, senior faculty member. But my lab's now been around for 14 years, and so I've been doing this for a while.
Yeah, that's quite amazing. And on the side, you've also been doing art, right? And I'm gonna pull this your Instagram, like freaking 34,000 followers on Instagram, and this is nothing to do with your science. Well, I guess there's some overlap, right? Like as far as this is obviously very nature-inspired organisms. Yeah, I
would say it's turned out to be the most effective form of scientific communication I've ever done.
Oh, all right. How is that?
It's if you start blabbing science at somebody, you can mean well, but it's not. People really have to already be ready to be engaged. But if you put something they've never seen before in front of them, that is just kind of intrinsically interesting, and then they start to ask questions, and then you say, yes, this is actually a real thing, and these details, right, are reconstructed from the fossil record and so on. And they're like, oh, tell me more about this. It just turns out to be this way of presenting stories that I never appreciated would be, orders of magnitude more effective in reaching people who are not themselves scientists already, right?
Let's do it then, because I think that's most of the audience here. So talk to me about the trilobites here.
Trilobites, yeah. So these are these ancient marine organisms, right? So they lived starting about 500 million years ago, 500 million years, and they went extinct 250 million years ago, right? And so if you're thinking like timescales, then 65 million years ago you had T-Rex, right? So we're talking like they're way older than the dinosaurs, and remarkably, and they have this sort of cool look to them. I don't know.
Oh yeah, they do.
I just think they're neat, right? And they're incredibly diverse. There's like 28,000 different species that have been described.
Oh, really?
Of these things. And
how subtle are the differences between them?
Some of them are truly subtle, as you can imagine. Some of them are they're just utterly different. Let's see if I can.
Oh, cool.
I think
Let's see. Is there another one here on the website?
Yeah. So if you scroll down.
Is this guy one of them?
That is, and the one on the right, in the middle there, the golden one.
This guy? This guy.
Yeah.
Oh, so these are two different ones than the first one.
Yeah. Yep.
Oh, that is so cool. And you see, this guy that we were talking about before the podcast started, this one's a real one.
That's a real animal that exists today, and also.
But it looks very much like trilobites.
That's right. And they're both arthropods. Okay. So they are related in some way, but they.
Wait, what's an arthropod?
Arthropod contains things like everything from flies and insects and bugs and stuff like this. Also spiders, centipedes, everything you think of as being a bug is an arthropod, but it also includes things like crabs and shrimp and.
There are some really tiny organisms that look like spiders. Are those arthropods?
Yes.
Like viruses and stuff like that, are they?
No. Yeah. So viruses themselves are not cellular. They're not animals. So everything we've just been talking about, they're animals.
Okay.
They're multicellular
Cool.
organisms, right? But viruses, even though they have those leg-looking,
It's weird.
They cannot walk on them.
Oh, okay. They just like latch onto stuff.
They're attachment structures. Yeah, and viruses are distinguished by this idea that they themselves do not have the machinery to reproduce themselves. They require a cell into which they inject their genetic information, and it takes over the cell and forces the cell to produce new viruses, which then explode out of the cell. Yeah.
Okay, so like this is one of the questions, I guess, that I had and that I want to ask you, and that I'll probably reiterate at some point because it's just a great interest of mine. But life, right? Like we were talking about also before this at some point about how life, to me, it seems like there is a threshold in which or by which non-living things conform a cell, and then the cell is considered a living thing. Does what the definition, does that just have to do with its reproductive capacity then, or?
I mean, mate, that's incredibly fraught territory. It's like trying to figure out essentially, what is life? Where does life begin? What is how is it defined? And I mean, I can tell you personally that I was incredibly inspired back when I was thinking about becoming a scientist someday. I read this book that has inspired generations of molecular biologists called What is Life, written by Erwin Schrödinger of the Schrödinger equation, right? So quantum physics kind of world. And he was grappling with these same sort of questions. I mean, literally the title of the book is What is Life? Question mark, right? And he was talking about, the production of negative entropy and all these, very big ideas about how you could kind of put a definition on these things. And I then sort of jumped off into looking at stuff that the Santa Fe Institute had done. There's a whole artificial life community that I found very inspiring. People, like, again, grappling with this sort of question of what distinguishes, exactly as you're saying, like what distinguishes biological systems from non-biological systems.
Right.
And it's sort of clear that like a protein is it's a biological molecule that is not itself alive, and yet the cell that contains it is sort of obviously alive, right? And resisted for generations. And so there,
But wait, why is the cell then obviously alive?
I mean, in the sense that if anything is alive, I mean, these things grow, evolve, they change, they
reproduce.
Yeah,
live, die.
And that gives you, I think, your instincts are very much like my instincts were, right? Which is there must be some way to kind of put these individual features together in a way that admits a definition that everybody can agree on. And
well, actually, that's not how I view it.
Maybe actually then where I ended up we're more aligned, which is that there's a bunch of people trying to do this, and you can never, it just ends up being some sort of semantic definitional argument. Maybe it's an interesting question, but it just seems to me to be some swirling vortex in which some people get trapped, and they, nobody.
It's
cares.
just arbitrary, kind of, right?
Right. Yeah, exactly. Like, is that what's really at stake? If you can say whether I can now have a decision procedure that says item A is alive, item B is not alive, right? Like, who cares? It's more
well, who cares from a scientific perspective, but say from the perspective of trying to understand consciousness, or what might be the minimum expression of what the universe has in terms of experiencing itself, then I'm very curious as far as, like, is there something like what it is like to be a cell? And then is there something like what it is like to be a part of a cell instead of just a cell,
Yes, in the Nagel sense of what is it like to be a bat, kind of thing, right? Like, what's the nature of experience? I should say that the executive director of the Computation and Neural Systems program, who is the guy who was my last interview the second time around at Caltech, is Christoph Koch, and he spent his entire career looking for the neural basis of consciousness.
That's a tough one.
Totally clear that, like, okay, let's set aside a set of belief systems that you can certainly have and I don't have, right?
Okay.
So imagine that this is what there is, right? Like, we are features of the universe. There's nothing beyond the universe. And
wait, what do you mean beyond the universe?
Meaning something that stands apart and causes the universe to do what it's doing. A typical deity type of thing. Like,
is that? But I mean, that seems like a flawed assumption to make. No, like as a starting axis.
You can say flawed assumption, right? Like, the question is, what can you do with the assumptions once you have them? Right? And I think one of the things that's been very surprising is that things that have, of course, I'm an evolutionary biologist, right? Things that seemed absolutely inexplicable without invoking some sort of deity, we now accept are absolutely producible by processes that are shockingly blind, right?
Surely.
And so that sort of,
Oh, the blind watchmaker thing, huh?
Yeah, exactly, right? Like people were just certain that biological complexity must arise, through processes that required intelligence, intervention, direction, and so on. And now, I can tell you from inside the field, it is abundantly clear that you do not need to invoke those sorts of ideas.
No, yeah, I agree.
Right, huge amounts of what happens. So, and that gets back to the assumptions. Like you can assume one thing or another, but eventually you're like, well, this is the role that the intelligent being, whatever it is, needs to play keeps shrinking over time. And so let's just imagine that eventually it will shrink to zero, right? Like we don't need it anymore. And so I'll just teleport to the end of that and say, that's where I am. As a working scientist, I'm working within the axiomatic framework, which is that we can, there is an objective world to be understood.
And we are separate from it.
No, we're in it. We're part of it. We're absolutely.
Oh, okay. That's the part that I was disputing.
Yeah. So we're phenomena that exist within it, and so it is feasible to understand. For example, ultimately feasible. We can't do this right now, but to understand, say, human consciousness and the experience that we have of being us, right? And you being you and me being me.
But when you say when you talk about understanding something like consciousness, you're talking about understanding it in a scientific way, right? In a scientific manner.
That's right.
Yeah, well, and obviously we can't do that now. But how can you understand consciousness in a scientific manner aside from understanding its correlates, say in the brain, and how it might function? Because from my perspective, you can never slice open a brain to find consciousness there, right?
Yeah, sure. But there's indirect measurements are we do them every day, right? We discover things about yeast cells that are not disputable, really, right? And
but when you say we discover it, you're discovering it also on the bed of consciousness. No, there's nowhere else to discover it, right?
So I'll dispute that even. I think so. One of the books that really inspired me in getting into, the graduate program in the first place.
Okay.
Was I spent a lot of time, like many of us do, right? And let's just stipulate here that, like, yes, I'm a professional scientist. No, I'm not a professional works on consciousness scientist, right? And I understand the distinction. So both of us at some level are occupying our armchairs here and engaging in discussion about things that interest us, but
it's fun. It's fun.
I don't want to pretend to have expertise that I don't. Right? It is fun. No, it's actually what wakes me up in the morning. Those big ideas and my team, right? Like they're just watching human beings do things very well is incredibly inspiring. But there's this book that you may want to check out, viewers may want to check out, which is it just really crystallized things for me. The book is called The User Illusion. Or Noratrander is this Danish science writer, and it just seems to be sort of out of nowhere that he takes this point of view of like sort of working up from thermodynamics and Maxwell's demon, and a lot of these very basic ideas, the nature of consciousness, and the thesis of the book. The subtitle of the book is Cutting Consciousness Down to Size. One of the things that he does very early is to say, what do we know about the bandwidth of consciousness that is relative to unconscious processing, right? So the work that we're doing upstairs here is not all conscious.
Sure.
It sometimes feels that way, but so he's like, is that really true? So there are ways to measure this, right? You can provide people with things to read, right? They have to read, each of the words consciously, and you do tests like that, and you can determine that the bandwidth of information that people can take in consciously is about 16 to 40 bits per second, I think is what he quotes.
Okay.
So then you can do experiments in psychophysics, which is a real word and a real, real people actually really do rigorous studies of these things, where, for example, you can present people with images 100 milliseconds apart, so fast that they cannot consciously tell the difference, right? That there are differences between these images in some
but
cases.
They're still registering them, huh?
They're still registering, right? So they can demonstrate that it's like possible for people to distinguish what has happened. But if you then ask them the question, did you detect a difference in the two images we showed you that you can't discriminate, right? Or were they the same? That people can answer those questions way higher than chance.
Yeah.
Right? And so you can accumulate and you can change those images in big ways and small ways and so on. And through those and many other experiments like it, you can determine that the bandwidth of subconscious processing is something like 11 million bits per second.
Nice.
Okay, so you have 16 to 40 on one side, 11 million on the other side, And this stuff, you know, the 16 to 40, that's evolutionarily new. Most of the stuff out there, it does not appear to have the kind of like processing internal monologue, right, that we have. That's relatively recent. Certainly yeast cells have no such thing. They don't even have a neuron, right?
So you would say there's not an experience of what it is like to be a yeast cell then?
Well, I think actually there's not as much of an experience of what it's like to be us as we think. Part of the
I mean that's part of the great mystery. We can't be aware of all of it at once, and we're processing so much more. I mean that's
totally.
It would be incredibly boring if we were to process all of it and we just knew exactly what's going to happen all the time, right?
But this is really the point that Neurotransfers makes in this book. And so let me give you the punchline, right? So he's like, you know, given that sort of ratio, right? There's this extra feature of consciousness, which is it is our picture of ourselves, right? And so he's like, by the user illusion, I mean something like your PC operating system, right? It appears to be populated with things like folders and files and your mouse and a cursor and stuff like this. None of that stuff exists really, right? It's the user illusion that is presented to allow you to effectively do goal-oriented actions at the machine code level, right? With this incredibly complicated box whose internal workings you truly don't understand, right? Like you and I don't know anything about the chips that go, you know, that actually make this stuff go. We require this user illusion in order to engage in this goal-directed behavior. So it's like, look, consciousness is the body's user illusion of itself.
Sure.
It's a system that this stuff in the back uses to operate the rest of us. And actually, a lot of this stuff that's going on up here, consciousness, is just storytelling that we do to ourselves after the fact. The decision's already been made, and then we convince ourselves that we've made the decision. So it's classic experiments from the 50s and 60s with Benjamin Libet doing these delayed choice experiments.
It's wonderful. It's so awesome.
So anyway, you get the idea that there's plenty to be explored here. And my personal point of view is that consciousness is wildly overvalued.
Overvalued.
Overvalued. It's reified. It's, you know, it's sort of.
But you see, the story isn't the story the most essential and the most important thing? Because without the story, there's nothing.
See, people feel that way. I don't think that's required at all, right? I
Really?
think if there were no conscious beings on the planet, the planet would still be incredibly interesting.
To who?
What do you mean? Why is
to what?
I mean,
because there would be no statement to be made of it at all. There would be no experience of it.
No, there would be the universe itself. I mean, what? It's like if a tree falls in the woods and there's nobody around to hear it, like, did it really happen? You're like, yes, it really happened. It totally happened.
That would be I mean, what about from the perspective of quantum physics? Did it really happen?
No, sure. We could go into does it happen in all possible worlds kind of thing, right? But certainly in some worlds it did, right? And if you're, what's the nature of reality? Also very interesting. But I do think that, you can take a position on this, and some people do, right? Like Sam Harris famously kind of has this, and I love his intellect. But he has this position of like consciousness is the one thing that truly matters all the time. It's the only thing. I'm like, no, I simply deny that. There's no, that if you push down into it, you're like, well, that's just an axiom, right? Like that's chosen. If you believe that way, then you completely do the thing. But I'm actually fascinated with all the stuff that's going on back here. That's where my ideas come from, right? Like take the free will discussion, right? Do we have free will? I think the answer is absolutely not. Is life still worth living, right? If you don't have free will, absolutely. Yeah,
you can get on a roller coaster and know that that there's an end to it, and still, be thrilled at the process, and
Sure, right? And then there's this, what is life for in the first place, right? Is it just to have a bunch of emotional experiences? I don't think so. Right? I think there's kind of making a difference in the ripples of the world, right?
So you don't think it's about the emotional experiences then so much?
No, I think happiness is a means, not an end.
Okay, well, unravel that for me, please.
That the reason why we have emotions, and certainly not all living creatures have emotions, right? Trees do not have emotions.
I don't know. I wouldn't know.
Okay.
You know?
Fine.
I'm not a tree.
There's probably something that maps onto emotions. But what is so? Okay, wind it all the way back. What's an emotion, right? An emotion is a modal state that allows a large set of repertoire, behavioral repertoires to be narrowed in a conditional way to a smaller set of things in a useful way, right? And so if you're, one of the reasons we like being happy is because it's associated with many options are possible. I don't need to narrow myself in right now, right? Being terrified has a very, very narrow kind of like fight or flight kind of Those emotions are
Oh,
useful.
that's interesting. Cool.
The reason why we have those modal shifts is because it has been adaptive for our species and every species that preceded us, right? To have these sort of modal states. And the issue is that, I mean, of course, every time, well, not every time, but because of that, many times when things are going really well, we are very happy. And so we start confusing one for the other. That somehow the going very well is not the goal anymore. It's the being happy. It's like, no, that's not the case, right? Because this stuff is just happening in the brain. I mean, if you really believe that being happy was the answer, we should absolutely be doing research on how to directly stimulate the brain to make everybody feel more happy, because then everything would be fine. But we've all been through that thought process as 12-year-olds, right? Nobody thinks that's actually a good idea. And the usual way out is like, well, that's not true happiness.
I see.
Right? It's not authentic happiness. What do you really mean is like, it's a mistake to do that. It's not what it's about. It's about the things that make you feel happy, right? Those things we can now debate about what should make us happy or not. But the happiness is a side effect and a motivator, and it's a means. Let's now talk about the end. What is the end that we're trying to reach?
Perfect. And how would you see then happiness? Do you think that sadness is necessary for happiness? Is that a necessary contrast?
Is evil necessary for good and things like this? I certainly think the contrasts help. Much like you live in Southern California?
Yeah.
Right. You grew up where?
Peru, Lima. It's warm in both places.
Okay. So, I lived, until I was 7, I lived in Boston. And then, as I said, I graduated from high school in Iowa. So you have all 4 seasons. And then Texas for 7 years, which is super hot. And then Pasadena, right? Southern California. And I know the Southern California thing. You just become very, very, inured to the idea that it is sunny and 72. Fire the weatherman, right? Like Chamber of Commerce Day every single day. It's fantastic. I don't know. I can persist in that state for an awfully long time. I really don't need winter to show up to remind me how great it is, right? I can remember, but it does help, right, to have those rhythms.
Like a baseline of sorts, right? Like some sort of idea of what it is.
You totally would lose track over an infinite amount of time, or like.
You need some semblance of a reminder at some point of what it is, the contrast that you're. Yeah. Some of those are just stories too, right? Like, and I don't mean stories as in they're not real, but rather that you haven't lived them yourselves. Like everyone's been told, like like eat all your food. There's starving kids in Africa, and it's like, oh, okay, well, like I should eat all my food, type thing, right? Because that's a very reasonable argument. So yeah, I'm with you. So basically, you're saying that the contrast helps, but isn't a necessity at all points in time, but rather
isn't fundamental.
Cool.
Right.
That's a very interesting take on that.
I think people get riled up about inequality, and I do myself. But it takes some forms that I think are not really valid. Like if some millionaire is upset about damage to their yacht. We're like, oh, cry me a river. You shouldn't be upset about that because you're a millionaire. It's like that's actually not the way emotions work, right? They're all deviations from baseline, right? And so if you start comparing your pain,
that's a mistake.
It's a mistake. Where you're like, there's somebody else on the planet who has it worse than you, right? So you shouldn't be complaining. No, it's normal to be human. It's normal to have your baseline. It's normal to be upset when that deviates, when expectations don't meet reality, right?
Yeah, I mean, if you were to try to quantify suffering or something like that, right? And what if a teenage girl, kills herself after she breaks up with her boyfriend, and then the kid that's starving in Africa doesn't? Then who's suffering more? It's it's not really.
And they're just people with worse problems than we have. We're able to have this conversation because things are going like reasonably well. It doesn't mean that we can't feel pain, right?
Yeah.
So that we can't be upset or weep about some loss, right?
No, yeah, I think everyone fluctuates to some degree.
Of course,
It's easy for me to try to conceptualize of human beings as some sort of like energy wave or just sound wave of some sort. Like just trying to think of like the ups and downs, the crests and the troughs and whatnot. So,
set point theory, right? The idea that like each of us has some sort of baseline emotional state, and some of us are fortunate enough to be like mostly happy most of the time, and some of us
is that a thing for me? I see like some pattern where it's like kind of like normally I'm just at peace a lot of the time, and then I have very exalted states where I am just besides myself, and then I have then I dip low usually, and I'm not sure if there's an order to it, but it seems to be somewhat of a pattern where it's like, and what I would think of as, say, depression, for example, is that lower part of the wave, in that sense, when you're just feeling low energy, right? Like exactly that, like when you just feel low energy, don't feel like doing much. Yeah. And if that extends for too long, then you start maybe getting worried and looking into it. But if not, like most of the time when if I feel depressed a given day where I wake up and I'm like, oh, I feel so low and like just so dim, right? I kind of just, I don't know, sit through it, and I kind of just appreciate it to some degree, where it's not a bad thing.
You're an enlightened being. I mean, I think that's the kind of mindfulness meditation sort of approach to things, which I came to very late in life, of just to kind of stand at a bit of a remove from your emotions. They don't need to have you while you're having them, and kind of let them, wash over. I think that's a talent that needs to be developed, right? It's not something that most of us, and certainly I'm an extremely emotional person, right? I'm swept up in these. I have exactly like you. I will say my set point that I return to an infinite time turns out to be pretty happy, and I feel very fortunate to have that. But I also know that that's not true for everybody. Everybody, there's some people who are just sort of chronically depressed, but then have times when they're able to be enormously happy, right? So it doesn't dictate your fate, but it does sort of say where you will settle down to. But like you, I then have these long range modes, like throughout a year. I mean, I came to StarCraft because there are times when I just work super, super hard on my science, or back in industry, like working on whatever project I was working on, pour myself into it 24/7, sleep under the desk, slide pizza under the door. That's all I need. And then I'll wake up one day and be like, I cannot do any work. I'm done. Stick a fork in me. There's nothing you can say to me that's going to get me to work. And I'm hopeless. And then for months, I'll do nothing but play video games. Right? I'll just sit there and everything is just, it's all game. And then one day I crawl out of bed and I go, I cannot play another video game. I'm done. I need to work. Boom. And so so I go back up. So if you average over a year, what I've learned now, I'm 47 years old, right? So I've kind of lived a while. And what I've learned about myself, at least, is that I can get very depressed if I judge myself for any one of those highs and lows, right? Or very impressed with myself if I judge myself for the highs. Really, I need to integrate over a year. And what that's allowed me to do is just say, roll with it a little bit, build my life so that it's okay. Like over a year, I can do some really impressive things. Like I'll knock your socks off, but right now I cannot do anything. I'm hopeless.
That's cool.
Being, right?
Yeah. That's a really cool way to look at it. I guess for me personally, a lot of the time when I'm feeling down or up, and with this mindfulness type approach that you're describing, I don't know that that's the way, where it's like, because it dims everything for better or worse, right? Where it's kind of like it's more stable at a center of peace, rather than really getting, really just feeding from things where you feel like God, or where you feel like the worst thing to have ever existed, so I don't know, like I find a lot of these ideas in as far as like meditation and mindfulness, like they're really cool for maybe a person that wants to get to a stable place where maybe their baseline, like you were saying, they feel like is too low and they just want to do away with so much anxiety and so much fear. But I'm not sure that it's for everyone, in the sense that sometimes I think it's just fine to just have like the really high highs and the really low lows, and not really, because when you don't judge them and when you don't, then you're not getting sold on them,
I mean, I do think that's fair. I think I just think it's like a tool in the toolkit. Right?
Yeah.
And of course, it's a safe statement to say it's not for everybody, right? Like who would say? I do think it's really interesting to, have the tool versus not, right? And be able to deploy it when you need to, but to be transported with joy, right? I don't want to stand back from that, right? Yeah. I don't want to notice it. I don't want to notice it, right? I just want to be it, right? Yeah. I want to inhabit it, right? I think that is, I'm young in meditation, so I'm sure there's people who are experienced meditators going, you idiot, like you're at level 1, right? And by level 2, you would realize that all these statements that you're making about meditation are silly, right? And I'd be like, I'm sorry,
Oh, no clue.
I need a coach, right? Exactly. But at least at this stage, that's I'm with you on the.
No, I think that's a really cool, really cool approach to just have the tool in the toolkit. I'm not sure if it's always if it's the best if you can get lost in things sometimes, and having that, not always. Yeah, you're right.
This idea of flow.
Mm-hmm. Yeah, right? I'm big on that.
Yeah, of course. It's getting lost, right? Like you lose track of time, and that's where my best work happens.
Mm-hmm. As well. Yeah, I think for most athletes, for most people that are invested in, that's Matt. That's Cobra Venom, the producer.
Hey, Cobra Venom.
I think
omnivore.
I think. Let's talk about Trilobites, okay? This is pretty cool. I'm gonna bring them back up. So, Trilobites are Trilobites. I can get away with it. I'm Peruvian.
Three Lobite. Yes, that's true. Drummond.
Wait, Drummond?
Drummond.
Drummond. That's so difficult. I thought it was French at first. You said it's Scottish?
Yeah, Drummond. Maybe it was French in the past, but it's Drummond. Yeah.
That's so cool. Alrighty. So a lot of these bugs, how do you even forge them, first of all?
So we live in the future, and that involves being able to have free tools. So I work in Blender, which is a software package that you can use to make Hollywood quality kind of movies. And you can see, look at blender.org and you can find any number of demonstrations of what they can do. But I use this to build models that are 3D printable. 3D printing, of course, everybody knows about, but just to kind of remind people, this is take a digital model and then produce by some sort of additive process, produce a physical version of that you can hold. It means that you can't, if you've looked behind the scene, rotated behind the scenes in something like StarCraft, you realize they're not built to be solid. They're just, they're sheets of polygons, right, with texture maps on them. So, for a 3D printed thing, you have to attend to making it a solid object that can support its own weight, that can, that doesn't have the surface painted on it, right? That really has all the geometry there. And that's one of the things that I've specialized in, is sort of making things that are very, very detailed. But
that's so cool.
The key bit of living in the future is that that you can now press a button and upload one of those 3D printable digital models to several companies that will then 3D print it in a jewelry scale castable resin, like investment casting kind of wax. And then they will cast it in the precious metal of your choice, or semi-precious metal, bronze, silver, gold, platinum, you name it. And then they'll ship you that piece of metal in a box. Of course, they charge you for it, right? But the fact that you can do that, I went from having dreams of making metal insects to going, I can do this.
That's so cool.
Suddenly you can do it, and there's a lot of learning, of course, to make them in the first place, to model in a way that actually will support investment casting. Right?
Do you use ZBrush, or what do you use for the modeling, the actual model?
I use Blender.
Oh, Blender is a modeling tool.
That's right. Yep,
okay.
right.
I thought it was like that's so cool.
Yeah, it's great. It's got a steep learning curve, but it's absolutely worth it, in my view. It has unbounded capabilities. I mean, I just scratched the surface of them. But I'm now, I donate to the Blender Fund to make sure that other people have free access to it on an ongoing basis.
That is so cool. Have you tried other things like ZBrush or like Maya or 3D Max?
Dabbled a little bit, but I committed to Blender early, and that turned out to be a very good choice. That's
awesome. Yeah, I mean, they look amazing, so I have no doubt. And the eyes here for this tarantula, for example, that's a different material.
Yeah. So then I got more and more elaborate. That's actually a jumping spider. Of the sort that would live on your windowsill, right?
Oh, no way.
They're tiny little things, yeah. But those are black glass eyes. And so I ordered those from a scientific supply company, and I built the model digitally around that. So that I mounted those eyes in the model itself.
So you mounted the eyes after?
Yeah.
Oh, okay. So it printed the metal, and then did you glue them, or how did you have them stick?
They're snapped in the sense that the modeling is so precise that I can just push them in from the back from
that
the
is
side,
so cool.
right? And they work. That was very satisfying the first time that those eyes snapped in.
I bet, yeah.
Especially because I had a show coming up, but yeah,
Nice.
That's one of my favorite pieces.
So this shows how often do you have them? Whoa.
You know, I've had one solo show, December 2019, and then I have my next solo show coming up in June, same gallery, Rock LaRue Gallery in Seattle, in this coming June. You know, world willing, right? We live in a very strange time right now. I had to cancel the show. It was actually originally scheduled to be in this past September. And then I had a piece that's all the way up at the top, that little strange plant hopper looking thing, was
this guy?
Well, not actually the one just below.
This guy.
Yeah. That one I sold. Well, that's sort of a belly view of it. There's also a video or that guy over there. That was the show in Australia and sold there. So some lucky Australian has him.
Do you care for sharing how much this go for?
Sure. I mean,
this guy.
I forget what I priced that at, but I think it was $2,000.
Nice.
And,
That's
part
so
of it
cool.
is that that gives you some sort of sense of the scale and details. That's my hand.
Yeah.
The process of producing them is so expensive, and of course, I don't charge for my time at all. If I did,
You don't?
No, of course, because I just do this for love, right? Like I'm a tenured professor, right? I run a lab. I have my day job is such that if I really divided my salary by the number of hours, right, that I work, and tried to factor that in, no one could ever afford these things.
Fair. But you should still price them up, just even so that
Well, I also want them to be out in the world, and also and yeah, I have been surprised. So I started out, the way I sort of turned pro was that originally I was just selling like little necklace style things, and those started to blow up on Instagram.
Oh, we saw one of those earlier, the jewelry one that I was looking at earlier. I want to find that one. It's so cool. Is it here somewhere? It is, right? Oh, there it is.
Oh yeah, that is a much more elaborate piece. That was in my show. That's a budding yeast cell. That's the model organism that we work on in my lab.
That's so cool.
Yes, but I had this dream of like making the kind of textbook cutaway, but with all the little details, and those are the outside there is 3D printed steel, the inside is 3D printed cast bronze. Inside of that, those little the jewels reaching toward each other, those are the chromosomes that are separating. So this is the part of cell division called late anaphase, where the mother cell, which is the big one, is splitting off from the daughter cell.
Oh, that's so cool. So this is the process that we're looking at.
Exactly. And each chromosome, and there are 8 of them, so there's actually some yeast species that have 8 chromosomes, is that apatite gemstone. Apatite gets its color from phosphorus, which makes up the backbone of DNA, right? And the DNA, of course, in a big long string is what the chromosomes are, and that's what we pass on to our children and so on. So the whole thing is meant to be a kind of, scientifically engaging, although not, exact in every detail. Of course, these are all sort of fluid, membranous structures that I've had to represent in some way to get them to go.
Because that's interesting you say that, because I was about to say, like, when I look at a textbook and you're looking at cells and stuff like that, it really doesn't represent quite as well as this might, like the complexity of the organism, right? Because it's just a flat, usually 2D image. Whereas this looks like, like an enhanced big version of more of like a living thing, but it also feels very mechanical in a sense, right? Where
there's a bit of that, right? The idea that I want to invite people to look closer, and the experience of looking at it on a screen is entirely different from the experience of holding it in your hand, right? It's like virtual reality without the goggles, right? It's you're just there with the thing, and you can move it around, and it has a presence that's not really possible in a photograph. And I do like it for that reason, right? I wish that I could teach people cell biology by having these sorts of models. These trilobites, the one that
this is the trilobite from earlier, but you've condensed it.
They roll up, yeah. That's the defense mechanism, right?
Oh, that's so brilliant, and it's worked into the sculpting big too, because it was bending around your finger here, right?
Well, that's just an illusion. It's actually one of the things that I'm working on right now is something that will fully enroll mechanically, in your hand.
Oh, these are just different.
That's
different.
Right.
Oh, that's so cool. So you modeled this one.
Yes.
Oh, that's so funny.
You like that?
Oh, yeah. Well, I fall for it. Yeah, I
Well,
like
good.
it.
That's perfect, right?
Yeah.
I love that little kind of the guy draped over my finger, completely exposed. But so
Yeah.
all those legs, what you see in the rock typically is just that top shell.
Uh-huh.
That's the classic trilobite that most people know if they've been, if they know something about fossils. And as every kid wants one. But if you flip them over,
Every kid wants one. I'm not sure about that. Maybe you definitely wanted one, huh?
I definitely, yes. Sorry, I should not generalize to all children. That's right. But I found that they're this kind of gateway drug for paleontology, right? Like people are just like super psyched about trilobites, and I was too, right? So this all started.
Why do you think that is? Is it their simplicity or? Yeah,
some kind of cool combination of the fact that they look like a Zerg drone. No, it's it's their symmetry, it's their simplicity, it's their you can connect with them because they have like eyes and you can see the front and back. They're not these weird foreign things, and they're not too freaky in the sense that I put in all the legs, and it turns out they had legs of that sort, and that's a whole other story of like doing the research to figure out what they really looked like underneath. But when you first see them, they're just like sort of
right here,
accessible things.
Yeah, it looks like something that I wouldn't mind holding as much as I would mind holding a thing that I don't understand as well. It's just like less complexity, I guess.
Yep.
That is so awesome. All right, so next show once again is June, July.
Yes, June.
Nice, June, and that's gonna be in your city.
In Seattle.
Seattle. Oh,
Rock
nice.
LaRue, ROQ LaRue Gallery, and that stuff is on my Instagram.
That's amazing.
Thanks.
Wow. Yeah.
It's been very fun. Like this was the Trilobite project actually started when I went through this super tough time in my life where everything just kind of fell apart. I was pre-tenure and wasn't sure how that was going to work out, and my relationship fell apart, and I just was completely devastated and knew that I needed some project that was not work and not my personal life, just something just for me. And so I went to the Field Museum and saw this incredible trilobite exhibits where they had new technology. Back in the day, somebody just took a hammer and a nail and cracked these things out of the rock, and so it would break a lot of the kind of structures. You could still see what was there, but it turns out now they have these micro sandblasters where they can just tease away the bits of rock around these very delicate spines and things like this, and you reveal these wonders that are just, they are aching to be lifted up out of the rock. And so I had this moment where I went, I have no idea what's underneath them. Is it like a slug foot or like, what is it? So I went and did a bunch of research and saw that under very special conditions, there are specimens that preserve all of the soft tissues, the gills, the legs, the antennae, right? And so there's real, actual, 400 million year old fossil evidence for these things. And I thought, this is my project. I'm going to reconstruct one of these.
Nice.
Just for me. And I posted kind of progress pictures on the Fossil Forum, which is a great site for like just amateur paleontologists like doing their thing. It's mostly people posting.
Is there other artists like that? Like, are there other people?
There's some artists. There's a whole section for people who do paleo art. Although mostly the site is excitement about dinosaurs and stuff like that. but anyway, so then I posted a picture of something on Twitter, and this guy from Nerdist, Kyle Hill, reached out and said, hey, do you have anything more like this? And I was like, no. And then I wrote back to him, I'm like, actually, I do have this other project where I've been doing this trilobite thing. And I pointed him at the Fossil Forum pages. And then, one day, January 10th, I think, 2016, I woke up, Saturday, I'm drinking my coffee, and suddenly, like, all my notifications start going off. And so Nerdist had written up an article on these things with some crazy title, like, biochemist professor reconstructs ancient trilobites by making them in metal or something, something that was like, sufficiently. And this very, very nicely written article illustrated with the pictures that I posted to Fossil Forum. And that thing went crazy viral, like
nice,
translated into multiple languages and so on. And so everybody showed up and they're like, well, what are you doing next? I'm like, no idea, right? Like, this was never the goal, right? This is just some little pet project. And so, that's one of those moments where life gives you the offer, and I said, okay, I'm going to take it. So here's what I'm going to do. And just started building the audience and doing more of these things. And and selling a little bit more. And then, after I sold a bunch of jewelry to people, then a gallerist reached out and said, would you like your own show? And I said, what does that mean? So, and then built the show, and that sold. So she said, do you want another show?
So wait, for the show, for how hard did you have to work coming up to the show? If it's your own show, how many pieces did you? Because it sounds like you went from making one piece to having a show. How many did you have to display?
I mean, it was one of the hardest, most stressful experiences I've ever been through. Just I had maybe a dozen pieces for the show. Some of them that contain multiple different animals, right, in one thing. Actually, one of the pieces was 11 trilobite species.
Oh, cool.
In different orientations, like in some array.
Mm-hmm.
And that piece actually was purchased by somebody in the Houston Museum of Natural Science, which is like a great thrill for me.
Nice.
They have a trilobite exhibit that is
so that's so cool. Right? Yeah.
So just amazing. So the people who know appreciate the attention to detail, and I appreciate that, and so that makes me feel good.
Of course.
But making those things was just killer. So I'm also trying to hold down this job.
I'm so sorry.
As a professor to run my team. I'm not sleeping. The 2 nights before the show, I slept a collective 3.5 hours, and spent all the time in a dark room, metalworking and trying to fix issues and fixing things that are broken and trying to put in packages. I've never done this stuff before. Shipped half of it, took the other half in a suitcase to Seattle. I've never had a show, right? And put it up, and astonishingly, I was working with a great professional on the other side, who knew exactly what she was doing, Kirsten. So she made everything happen there, but now I know I need like 6 months to make a show.
Yeah, it sounds more like it. But at least you must have a lot of these pieces as well that you can just take to your next show, or is every show?
It's got to be all new.
Oh, really? Every time?
Yeah.
Oh, wow. So what is your next show going to be about then?
Yeah, I'm just curious. Do you get to just show reruns of your best games? Like
no, not usually, no. Exactly. No, but I imagine when it comes to art, like you can just reuse the pieces that you still have.
There is some of that, but not for a new show. Then the opening has to be, because it's exposed to collectors, and the collectors then,
I see.
They want to see, what have you got that's new, and they want to have something that nobody else has access to.
That makes sense. Is some of the new show on Instagram yet, or is it not revealed yet?
No, and in general, the gallerists don't want you to reveal
makes
the final
sense.
pieces. But I will tell you that I'm doing a daddy long legs, right? A daddy long legs,
cool.
such as you've never seen before, right? And that last piece, actually, her body is here in front of me, right?
Oh.
I don't know if you can see this, but
yeah.
Right.
That is so cool.
That's straight off of my 3D printer. Right, and she's gonna have big long legs. She's gonna be about a meter wingspan.
Wait. So again on the process, you have the thing 3D printed now, but you have to make it into metal, and that's another 3D printing into metal.
So there's 2 ways to do this. One is to seriously, I still ship this off to a company and say, please, give me the metal pieces in a box. And these are now, the pieces that I'm making are elaborate. So they're multiple different pieces, and they have to be assembled in a certain way. And then I do all sorts of metal finishing and polishing and grinding and so on. But do
they snap casting, together, all of them? Like
many of them I have snap fit. Some of them I have, you put them together. I mean, I guess I can show you a little version of this this thing that you saw, the leaf hopper.
Oh, that's so cool.
Before, right?
Yeah.
So these are unfinished silver wings, and you can see they're just, there's a little screw there that's holding them in, and then the head comes and covers that up.
I see.
There's a bronze base, and the body has a top and a bottom. Those are actually snapped together.
Well, this is where you're engineering, when you're where you. That's so cool. So you're like combining all of your passions at once.
So there's the bottom and the top of the body, and those things will just sort of. I know this is impossible to see in this.
Geez,
format,
right? So the person that needs the person to do this would have to be an engineer and a biologist and an artist, and you just happen to be all 3, right? Like it's
it's the reverse, of course, right? Like you try to find some niche where you can do something that plays to your strengths, where you have some competitive advantage, and that I feel like if I didn't do this, these things wouldn't exist.
Yeah.
And I love that they exist. At some level, I'm doing it for me. I'm very I love seeing how you, respond to these things. That's meaningful to me, right? Like, and so I think that experience of shared beauty and interest.
The story. So you're saying the story is more important than the physics of things, and that's what you're saying.
Sure.
Yeah. All right.
I have
Well,
to say, like, I mean, of course, nobody. You sort of set this up as you picked me out of midair, and for all I know you did, but I followed you for years, and one of the reasons that I'm here and not on some other place is that I just find your approach to the game, your approach to gaming, right? The reputation that you've built, your insights just to be very compelling, and I like the way that you're, I said this to you before when we first met, right? So this is the second time that we've ever met.
yeah.
That it's obvious that you're a mind questing, and that you would like to think deeply about everything, right? And really challenge assumptions and get to the bottom of things. And there's a bit of the scientific ethos there, right? And I think something deeper than that about being a human being who cares why things are the way that they are, asking whether they need to be, asking whether you can personally make a difference in making things a different way, right? That might be better, more useful.
Yeah. Thank you very much. And I really, like I was telling you the other time too, I really, like for me, it's not about so much placing importance on trying to figure out like what the meaning of existence might be from a single angle, but rather just see how it all fits together, right? So there is an angle in biology, there is an angle in physics, and I think that whatever we tend to and we pay most attention to ends up being what we consider the most real thing, like because that's what we understand best, and life is just so beautifully complex that any angle that you look at it from it's gonna make sense. You know what I'm saying?
Yeah.
So
do you know about the idea of emergence?
No. Or I have a vague idea, but I'd love for you to.
Vague idea. I mean, I think this is a really fun framework where you can think about these kinds of things. Certainly has been very useful and inspiring to me, which is, of course, there's biology and there's physics. And they're not really two different ways of knowing about things, right? They're both science, for starters. But there's a very clear sense in which biology sits on top of physics.
Sure.
And that there is a reductionist idea, right? That you should be able to understand the things that happen in biology in terms of the physical processes. And so then there's this question of like, is that all there is? Is it just reductionism all the way? We just sort of pull the pieces apart and then we understand everything. If we understand the little pieces, we understand the whole. There's an amazing article by Phil Anderson called More is Different. It's a super inspiring article that talks about the ways in which this is not true. It's you cannot understand the workings of large-scale systems by understanding just the pieces and parts, right? It's insufficient. You actually have to do extra work to go connect things back. And this is the idea of emergence, right? So the idea of more is different. The action of a bunch of water molecules is not the same as something that you would appreciate from a single water molecule. A single water molecule cannot freeze. A single water molecule cannot flow. It cannot drip. It cannot splash. It cannot, it doesn't have surface tension, right? There's all these phenomena that are obvious, right, in large collections of water molecules that have no reductionist component, right, in terms of these lower level systems. And so what this creates is, first of all, the need to go beyond reductionism, right, and to think about these emergent phenomena. How many, you can ask questions like, it's not really well posed, but how many water molecules do you need until you have water?
Right. Like the liquid that we understand that splashes, etc.
Right? When you start to get these behaviors, where do those behaviors come from? Why do they exist, right? Like what the water to ice phase transition is a property of multiple water molecules talking to one another in a lattice, right? That doesn't exist when you have just a single one. So they're well-posed questions. These are scientific questions. But it also creates an insulation between these layers in which phenomena that exist at the level of, say, water droplets doesn't really have a corresponding deeper thing that you need to know about. You can appreciate and study the system at this level. And biology is like 12 layers of emergence now.
I see.
Above this, right? It's not just built on water molecules, it's built on the droplets of the droplets, in the structures, in the membranes, in the environment and ecosystem, and so on, over billions of years, right? That sort of those layers of emergence, I think, give you the sense that it's not hopeless to think that everything can be reduced to physics. It's far more interesting than that. And that there are these different, what is the right level of description for attacking some particular problem? One of the reasons I gravitated toward StarCraft was that it involves large armies. And the synchronization and orchestration of movements that have no equivalent in a single unit. You have to think at this emergent level. The strategies that you're playing. I mean, I can't play to save my life, right? But like the things that you're doing represent an intuition that is not at the level of a single Zergling, right? Like they are movements and trends and patterns that I would love to understand better how you think about these sorts of things, but it's clearly an emergence type of phenomena.
And consciousness is that from your perspective as well?
Oh, I think that's guaranteed. Right.
Yeah. That's very interesting. Yeah. I think that's a good way to conceptualize of things and to think about things. So, yet still for a physicist, they would be able to understand most of the interactions that particles at a base level or some of the most microscopic levels, I guess you could say, interact with each other, right? And from those mathematical ideas, you could then derive how water might work. And
you should
so
be able to see, right? Yeah, this question of is there new physics, right? This what physicists like to talk about. As you move to these complex systems, is there truly new physics, or are there new ways to put together the old parts, right? That we might not have previously appreciated.
Oh, is this a question that's unanswered then?
Yeah, right.
I see.
It's pretty rare, I think, for people to find new physics, and instead, mostly what they find is that there are new principles that can be put together from the lower level parts, because the space of all possible behaviors, right, of a large army, for example, right, that space is exponentially larger than the space of behaviors of an individual unit. And so it's no surprise that there are higher level patterns that exist, but they don't require knowing anything about, there's no new unit move, right? That would be new physics, right?
Yeah.
Something I had no idea that they could spout acid or whatever.
That makes sense. All right. Let's see. So another question that I had for you, and that you were mentioning, your mentor, he was Nobel. She was a Nobel Prize winner, and I believe it was for evolution, for like basically induced evolution. I'm botching the idea, but
directed evolution.
Directed, yeah.
So her name is Frances Arnold. She's still very much with us. She won the Nobel Prize 2 years ago in 2018 in chemistry.
That's incredible.
Yeah, she was my graduate advisor. She is an absolute powerhouse, a complete inspiration. It's very fun to be mentored by her.
How involved were you? How much, could you tell us about what this means? The, directed evolution. What is this?
Yeah, I mean, the essential idea is that, of course, things evolve, and there's a process of, I think the evolution that most people are familiar with is this idea of, species changing over time, animals changing their morphology, right, their shape, their behaviors over very, very long spans of time. What of course is happening in order to make this true is that their internal molecules are themselves evolving, and that process of molecular change happens typically at the level of, there are genes, right? So what a typical gene encodes something called a protein, like an individual molecule called a protein, which is a chain of amino acids. So when you think about amino acid, I don't want to, you probably know all this stuff. I don't want to insult you, but I do.
No, please insult me, because even if I catch some of it, I'm sure there's people wondering. So it would be good. Yeah, I understand. I don't understand. I think amino acids are produced by a biological.
Yeah. So these are words that we're familiar with because they show up in diets and supplements and things like this, right? So gene would just be a little stretch of DNA that you would have in your DNA or bacterium's DNA. The gene encodes a protein using the genetic code. So there's this idea that the individual nucleotides, which are the bases, the A, T, G, C, right, in the genome, is read out or transcribed into messenger RNA, which is basically the same type of thing, except it's instead of being a double helix, it's just a single polymer.
Half of a helix.
Right? Yeah, it's half of a helix. Exactly. That's a great way to say it. And that thing is AUGC instead of ATGC, but that's basically the only major difference that you need to worry about. Then that messenger RNA is translated into proteins. Okay, that is
by what?
There's a structure in the cell, many structures actually, often tens of thousands of them, called ribosomes. And the messenger RNA is fed in one side, comes out the other side, and then there's like a ticker tape. The growing protein will be read out in groups of 3 of these bases. So every 3 bases corresponds to 1 amino acid, and there's a specialized molecule that brings an amino acid in, has one stuck to its tail end, and it goes into the ribosome, sticks down on its 3 anti-nucleotides, anti-codons, brings out that new amino acid that gets stuck onto the growing chain, and you do that a bunch of times, and that thing comes out of the ribosome as a finished protein chain. So it is a chain of amino acids in just a very particular sequence that's dictated by the gene. Okay? And then that thing folds up into a 3-dimensional structure to some sort of, you've probably seen pictures of these things, weird ribbons and helices and things like this.
Yeah.
That has, the shape of this typically determines the biological function. So hemoglobin, right, is a protein that you have in your red blood cells. And hemoglobin is just a protein molecule. It's actually a tetramer, what's called. There's four nearly identical things stuck together in a group of 4. And hemoglobin has a job. Its job is to bind oxygen. And the way that it does that is by being in a particular shape, right? Having this long chain of amino acids adopt a particular shape that allows oxygen to bind and to unbind in a regular way.
Oh, that's cool.
Okay?
So does it have to do a lot with shape and not just components, or is it about structure, kind of like
shape? It's chemistry, right? How do you get if I just gave you, if we wanted to sit down,
kind of like how you snap the eye on your spider.
yes, or making a necklace, right? It would be the sort of easy way. If you and I sat down and just like put a bunch of different colored beads on a necklace, that would look like a protein sequence, sort of. The difference is that necklace wouldn't, spontaneously fold up into anything. And these things do, what 56 Nobel Prize, I think, was given to Christian Anfinsen for determining that it was just the sequence of the protein all by itself. Didn't need any of the stuff and other stuff in the cell. You could just take one of these sequences, drop it in water, and it would spontaneously fold up in this origami way. It's like self-folding origami. It's crazy.
That's cool.
The protein folding problem is the way that people refer to this. Like
how complex is the folding? It's like compared to the protein in itself.
Incredibly complicated, right? So
that's so cool.
The folding itself is, you can sort of work it out. You can trace the backbone of the.
Can you predict how it's gonna fold?
That's the protein folding problem. Because we know in a lot of cases that sequence should be sufficient to determine the thermodynamically most favorable folded state. Okay, and yet, and nature solves this problem.
Right, yeah.
Microsecond. Right? So it's clear that it's a solvable problem. But then people have devoted their lives to trying to predict the folded structure from sequence. So if for anybody interested in this, look up David Baker at the University of Washington, who is arguably the kind of leader in a very large field of people thinking about this protein folding problem. I have a colleague, Tobin Sosnick, who works on the rules, right? Effectively, like how can you efficiently and effectively determine what that folded structure is, and then how can you go farther and determine what that function is associated with that shape, because of course the shape itself is just a means to an end. And then I guess that's the
basis of
there, right?
Directed evolution.
That's right. So that's where we're going. So the background here is, add one additional bit here, which is that those genes that started this whole process of making this protein that does something, those genes are constantly being, randomly mutated by just mistakes in the copying, right?
Oh, what? Wait. Can you call them mistakes?
Well, no, I get you, right? Like it's a deep, it's a deep question. I've made a considerable amount of kind of scientific hay out of saying, things that look like errors may not be errors.
Right.
fine. So let's just stipulate that all I mean by mistake there is something that is different than what's encoded, right? So it's as if you had somebody copying out something that you'd written, and they write something that's supposed to be a copy that differs from what you gave them.
But do you know that it's, but we don't know that it's supposed to be a copy, is the thing.
That's right.
Right.
Well, we do. I mean, we do. Supposed to be. Okay, let's just now also teleologically in biology, it's always very tricky. I mean, these are really just physical processes that happen. But it is clear that like the main thing that happens, right, is that everything gets copied perfectly. So, you know, the error rate of DNA replication in E. coli is something like 10 to the minus 9, right? So like 1 in every 100 billion base pairs is mutated. Or, yeah, something like that. So 10 to the minus 9, 10 to the minus 10 is the number. And if you do the math on like how many E. colis you can produce, right? The answer is something like, you know, you get one change, let's say, we're not going to call it a mistake, but one change every, I don't know, 1,000 E. coli. Okay, so the vast majority of the bacteria are produced without any changes, and so that gives you some sense of like, well, probably the purpose of this thing is to maintain information, not destroy it.
That makes sense. Is that evolution in itself, though, if there is such a change?
Well, so that's the thing, right? So what is evolution? Evolution is not just the existence of the change. That change we call a mutation. The mutation.
Mutation is kind of like is that kind of like saying evolution but in a different light?
No, it's one aspect of so you know, evolution is not only mutations, but then differences in fitness that arise from those mutations, right? And the heritability of those mutations. That gives you natural selection, right? This is one of the things that Darwin is famous for. You know, you sort of turn the crank on those things, and then you'll get evolution, which is change in gene sequences over time. Right? Sustained changes. So if these changes just occurred and then, you know, then vanished, then we would be in stasis, right? Occasionally these little changes would rise, but it wouldn't do anything. There would be no sort of arrow of time, right? But instead what we find is that these things accumulate, right? And some mutations then cause the organism to be way better at eating a particular type of sugar, which means that everything that has that mutation suddenly can proliferate while its evolutionary competitors are left to starve, right?
All right.
So you can see there's this like link between the genetic changes and the changes that are made to the protein and that then give new capabilities to the organism. This is why, I mean, you know, StarCraft is filled with all of these cheesy, you know, mutations and evolution complete and all of that, right?
Yeah.
But, you know, the idea here is it's much more undirected, right? You don't know what you're going to get every time.
And and your mentor had a phrase for that, I believe, which was you get what you select for, I want to say, or something of the sort.
You get what you select for, yeah. So where does directed evolution come in? So that's evolution, right? So this change over time. And her big idea was, you know, everybody at the time was trying to make new molecules, make new protein molecules. Proteins are incredibly useful. I mean, they're like these little nano machines that can accomplish all sorts of interesting chemical tasks. She's an incredibly powerful chemist, right? So she's been thinking about like new reactions and how can you get things that, you know, how can you tame those reactions and make them practicable, right? As a chemist. And her recognition was that, you know, hey, many people recognize this, of course, that biology does this enormous array of reactions and does it incredibly cheaply at room temperature with no poisonous byproducts, you know, in many cases. So what an inspiring source of variety. So of course you go out, you know, you want enzyme cleaning action in your laundry detergent, right? An enzyme is just another fancy name for a protein. It's a protein that carries out a chemical reaction, okay? So you've probably seen enzyme cleaning action. What is that? It's a protein that's been added to your laundry detergent so that this protein, you know, what it does is it goes and
deconstructs dirt.
Chews other proteins up, yeah, like blood stains and things like this. Your blood is just hemoglobin, so it cleaves hemoglobin and gets it out of your clothes. Well, that thing's a protein. That protein, what is that protein? In many cases, that protein is called subtlysin. It's, you know, crazy name, doesn't really matter. But it comes from a soil bacterium. It's just a protein that soil bacterium makes. Now, sadly, that protein won't work in your laundry.
And that's if it's something that just to be clear, if it's something that a bacteria makes, then it is an enzyme. Is that how that works? Okay.
No, just an enzyme is a generic name for a protein that does catalysis, right? That's
okay. That has a chemical reaction.
Chemical reaction, that's right. Takes in what we call a substrate and turns it into a product, and then that's a chemical reaction. The idea behind a catalyst and behind an enzyme is that the enzyme itself is not consumed in the reaction, so that it can do many of these reactions. It's like a little machine where stuff goes in one side and comes out the other side.
Nice.
Okay. So it would be great if we could just go out and make a bunch of subtle lysine and just dump it into your laundry. But unfortunately, your laundry is hot, and it contains bleach often, right? And that will completely inactivate what we call wild type subtle lysine, that's sort of naturally occurring subtle lysine. Well, what if we could use the methods of evolution to make changes in the genes, right? And then test the effects of those changes and unnaturally select, right? In a directed way, proteins with new capabilities where ahead of time we can't predict. And there is no, the rational principle is not about trying to guide the structure of the protein in some way. Instead, the rational principles are about how to construct the evolutionary scenario that will, and screening process and so on, that will give you the molecule that you want at the end. That's directed evolution, okay? It's taming the evolutionary process for human ends. And that's what she pioneered, right? She invented the field of directed evolution along with
and when she said then you get what you select for, it was a little bit of a, it was more like, you don't really.
Yeah, no, so why is this difficult? Because in the end, okay, let's say the selection went as follows. You and I are practical people. Okay, we've just met. I have no idea if you're
practical. I'm not that practical,
You strike me as a practical gent.
Thank you. Thank you. I'll strive to be.
And so let's imagine, how would you do this? You mutate some gene, you put it in a bacteria, and you ask it to produce huge amounts of subtle lysine, okay? And then you harvest that, you purify it, you put it in you put it in some laundry detergent, you do a load of laundry, and you see whether your clothes are cleaner, okay? That would be the correct way to do it.
Okay.
But that's incredibly expensive. What are we going to do? Spend, multiple PhDs,
have like a million, million of machines running at once?
Yeah, exactly. No. So you're like, okay, what's the essence of this? Well, let's just say what I want to do is take a particular substrate that I can measure the cleavage of. Okay, I can measure whether or not subtle lysine is breaking this thing in half, for example. Okay, and I can put that in a test tube, and I can now ask. If I do that same reaction at slightly higher temperatures in the test tube, and I add in a little bit of bleach, are these new mutants going to be able to do that reaction? Okay. Now we're doing a selection with a screen, what's called a screen, right? You take everything that comes out of your mutants library, and you go, okay, folks, let's just see how you do, right? On this particular test. What she means by you get what you select for is, okay, at the end of this process, you have something that can carry out that test tube reaction. That's what you selected for, right? Provably, you have gotten that. But that's not actually making laundry clean yet.
Right.
Right? The hypothesis is then you've been clever enough, right? You haven't cut any corners. You've given it a realistic substrate. You've used realistic concentrations of bleach. There's nothing special about the tubes that you did this in. There's no other effect from cotton that happens to interfere with this. All these assumptions that you've made to try to, simplify the process so you don't have to do 10 billion loads of laundry. Exactly. Now you go back in, you do a load of laundry, and you find it doesn't work. Oh well, right?
Yeah.
Because you got what you selected for, but you didn't select for what you wanted.
Okay.
So this is the trick of oftentimes people will select for one thing, and what they'll get is a fabulous test tube degrading, enzyme that has no effect on laundry at all because of some other thing that they weren't selecting for the right stuff.
That's cool. But you do find stuff out through the process, regardless of what your initial aim is.
And part of the artistry here, and part of the intelligence, right? And part of one of the things that she did very well. She's very judicious about. She thought very deeply about the principles. Actually, that's why I joined her lab, as I took a course from her called "The Design of Biological Molecules," and it rewired how I thought about the evolutionary process. And I was like, I must learn more from you, right? This is very special. I've actually tried in my own courses to give my students, right, the same kind of aha moments about what it's like.
Can you give us a hint of though?
Yeah. So, imagine what it's like. You're in graduate school and you're starting a course called, day one is design of biological molecules. What kind of material do you think that the professor would start talking about, right?
Biological material.
Yeah, take a moment right in your head and just imagine what that's like. You're like, okay, I'm probably going to see some equations, and I'm probably going to have to some definition of what design is, and I'm probably going to have to, what are the molecules we're going to be focusing on, okay, and a review of the syllabus. Where she started was, please read this short story called The Library of Babel by Jorge Luis Borges. And the story of The Library of Babel is about people who are trapped in an infinite library, a library that contains all possible books. Inside all possible books are all possible stories, okay? Written in a language that nobody can understand. You can understand the characters, but they don't seem to make any sense, right? Because it turns out they come to slowly realize over time, locked in this endless labyrinth, that they're all randomly chosen, and there's no organization to this library at all. Okay? And occasionally one of these librarians will open a book and find a passage that's almost understandable.
Okay.
And this fragment of wisdom, and then everybody will come from hexagons around to try to determine what this might mean, and to try to interpret these stories. They're thinking about why does this exist, and so on. We're embedded in this sort of intellectual kind of, okay, what's going on? Her point is this is like the space of all possible genomes, where the stories are written in a language that's extremely difficult to understand. Random perturbations can change the meaning of these things. We struggle to think about the size of the space, right? If you think about how many different possible proteins there are, right? Of a typical protein is 300 amino acids in a change.
We're trying to discover still proteins, or
it's not just discover. We can't even scratch the surface of the set of all possible proteins.
I see.
Nor the set of proteins that have ever existed on Earth, right?
So we're making new ones then, kind of easily then.
We're making new ones, and one of her points here is that we have access as designers in protein space. We have to think about the space of all possible things, not the very tiny sliver of a rind of an edge that's been explored even by all of natural evolution on this planet since the beginning of life. That's a rounding error, right? On the space of all possible sequences, right? On the space of all possible stories, right? That you can write. You can do some quick math that says, how many protein sequences of length 300 are there? And there are 20 different types of amino acids. So 20 times 20 times 20 times 20 times 20 is 20 to the 300.
That's a lot.
It's not just a lot. It's more than there are subatomic particles in the universe.
Jeez.
Right? Right? It's just an unimaginably large number, right? And every one of those, in principle, could do something. So how do you explore that space? How do you even think about that space? That's the experience where, I'm sitting in class going, like, okay, right? Now you're in the presence of a mind.
That's cool. So how do you go about it then? Like, how do you? And is this the reason that we think of evolution then as random to some degree? Is there some connection there to the randomness of?
That's right. Back to the mutations themselves, right? The kind of underlying assumption, and there's all sorts of work on the source of mutations, right? But I think it is useful to think about the kind of usual way that we think about this, which is the DNA polymerase, which is the molecule that copies one genome and makes another genome, is just chugging along, and it occasionally makes a mistake. And it makes a difference, right? And that can be because a cosmic ray, hit at exactly the wrong moment and caused it to do. And it's also just because it's impossible, thermodynamically impossible, to make a perfect copying machine, right? It just, there exists some error rate. You can try to push it as low as possible, but one of the very interesting things about evolution, right, is that it doesn't make things perfect. It just makes things good enough. And one of the things that it can make, pretty good but not perfect is just the fidelity of copying DNA. Okay. So the notion is that the polymerase has no idea.
What do you mean by perfect though? When you say
perfect meaning no differences from the template.
I see. Okay.
Right. If you wrote something and you say,
but wouldn't that just result in like everything being the same thing?
Exactly, right? So I mean, these phrases, there's two ways to think about that, right?
Yeah, that's more perfect.
That's absolutely like nothing would evolve. That's not perfect at all. Exactly, right? So there's this notion of maybe these these mutations have to exist for reasons of evolvability, right? That's, I mean, at some level that must be true. Then there's this question of whether the mutations themselves have any bias in them that reflects a prediction about whether this particular change will be useful, right? And there, I think, you know, there are people who argue about this and argue about the different scenarios, but it's fair to say, like,
seems randomish.
Yeah, that's right. There's no strong evidence that there's bias mutations that can see the future. Okay, so what that means is the mutations are raw material. Without them, evolution doesn't happen, but they are not themselves evolution, right? So what causes a mutation to then be what's called fixed in the population over time. That is where it takes over the population such that, you know, all of us have, you know, the
same mutation,
exactly the same mutation, and everybody else has gone extinct. That's the process of the conversion to fitness. And there are all sorts of different ways to do this, but, you know, gene to transcript to protein activity to, you know, activity of the cell to success of the tissue to successful organism, you know, that's the stuff.
Okay.
So the mutations are random, but selection is not.
Right. And when we say random, obviously what we're saying is there's just such vastness that we wouldn't be able to wrap our heads around it or wouldn't understand it, or is it actually is there such thing as a random?
Yeah, I mean, you know, what does it mean to be random? But I think the idea is that there is no information that mutational process has about the future, right? That's what random means, really, right? It's, of course, they're not random in the sense that there are biases that exist. You know, it's much more likely to make a G to A mutation than it is to make a G to T mutation. That just has to do with, you know,
but if we could predict which one it would make. Like with certainty, would we still call it random?
Random, yeah, and that's what I mean, right? Because it's random with respect to fitness.
Okay.
That is, there's no sense in which the mutations that arise are the ones that are most likely to make us better. In fact, the vast majority of them make us worse.
I see.
Worse in the fitness sense, worse in the sense of being able to pass on genetic information to future generations.
Interesting. That makes sense. That's pretty cool. How do we know? How does nature know then to keep the good ones? Like, because they reproduce? Is that it?
Good. That's the word "good" there is doing all the work, right?
Yeah.
Nature doesn't know, and in fact, the only definition of good that we know is it stays around, right? Like whatever lives. I think there's, you know, it's the, you know, we talk about fitness and survival of the fittest and things like this. And I like to think, you know, one thing is a predictor and one thing's a result, right? So you can have a biased coin, right, that flips heads or tails, and it's, you know, 60% of the time it's heads. But, you know, you could have a biased coin like that, and you could flip it and get 10 tails in a row, right? The coin's still biased, right?
Right.
But you will just get those sorts of things by chance, right? So the underlying property of something being more fit or not, or having a selective advantage. That is, if you took this organism and you put it in an infinite number of competitions, it would win the vast majority of them. But still, on this one instantiation, it may lose. It may lose for reasons that have nothing, that are no more interesting.
And the mutation does not persist.
Statistical fluctuations. Yeah, exactly. Then the mutation is lost. So you can show, we know actually that even beneficial mutations that arise must arise
seemingly beneficial, I guess.
Well, I mean, we know they're beneficial in the sense that we can, in the laboratory, carry out the experiments where we can show that actually they confer a growth advantage on
animals, that's
right?
cool. That's cool. I see.
Right, but you can show that the probability that those things that arise are going to be lost in the next generation is, you know, most of them are lost because they only arise typically in one individual, and that individual is in a population of thousands or tens of thousands.
That makes sense.
Right, and so they can just, like, by chance be the one who just, like, gets stepped on. You know, doesn't mean he wasn't more fit.
Gets struck by lightning, and
right?
Who cares about your fitness?
He was technically more fit, but there's more to evolution than just fitness. Turns out there's also random effects, right, or what we call genetic drift and genetic draft and hitchhiking and all these other, you know, the subject of evolution, molecular evolution here is vast and wonderful and not reducible to this, like, oh, it's more fit, right? So
that makes sense. That's pretty cool. All right. Well, this has been super exciting so far. I would talk to you for hours, to be honest, Alan. So
I'm in your hands.
yeah. All right. Well, let's keep going then. But this was about mostly your mentor. What are you working on right now? Like, what's your most important pet projects or something that you think would be worth or interesting to share that maybe we can wrap ourselves around? Yeah.
Sure. So, my lab studies how cells sense and respond to their environment, and we're basic scientists, fundamental scientists, right? That we ask questions that are not, I'm not going to cure a disease tomorrow. I'm not going to make a new product tomorrow. I'm interested in how cells work, and the idea is that if we can learn kind of the principles by which cells do their thing in a generalizable way, that is the platform on which many of these more practical discoveries are built. So this question of like, how do cells sense and respond to their environment? The specific question that we spend a lot of time on is, how do cells sense and respond to temperature? So we know that they do. There's something called the heat shock response, where if you suddenly heat up a yeast cell or our cells, actually all what's called eukaryotic life, right? So things that are not bacteria and not archaea or eukaryotes.
Well, what are archaea?
Archaea are another branch of life that are not bacteria, not eukaryotes.
Oh, are they related at all to us?
Yes, yeah. So, I mean, at least so far as we know, all of these organisms are related by common descent.
Okay,
but
cool.
They're found typically in, they're often extremophiles. They're found in hot springs.
Cool. Okay.
I myself do not study archaea, but they certainly exist and they're a huge kingdom. So across all of these cellular, in fact, all cellular life, if you raise the temperature outside of the range where the cells typically are, let's call it happiest, they're able to proliferate and grow and survive. Right? If you raise the temperature a little bit, they don't like that at all, and they start to produce a response internally where they turn on a bunch of genes and turn off a bunch of other genes. And they express proteins whose job, enzymes and other things whose job it is to kind of what appears to be clean up a bunch of messes that are suddenly sprouting in the cell.
Like try to stabilize it.
Clumps of stuff that form in the cell. They're like, oh, okay, let's stop that from happening. Let's undo the clumps. Let's like kind of try to resurrect the proteins that get stuck in these clumps.
Cool.
So we're interested in a couple of questions. How do cells sense temperature in the first place? So it may be amazing to you that to this day, we do not actually know how cells sense temperature.
Yeah.
We know, for example, like in your skin, there are temperature-sensitive channels, right? So ion channels. So they're like protein structures that exist in the membrane that when the temperature goes up, they pass ions through them, right? Like
that's cool.
Small charge molecules.
And that's what connected to the brain. Where do we receive that information in the brain?
Exactly, cool. Right. So it's the channels themselves can be. Well, there's feedback, but they themselves are typically on nerves, for example, so directly change conduction right into those nerves.
Cool.
And we don't know how those channels actually work. Right? We don't know how they sense temperature at the molecular level, which is we don't have like an atomistic description in this reductionist way that we were talking about before of like how they sense temperature. And yeast cells, basically, I mean, they do have one of these channels, but it's not required for any of these responses. And they're perfectly able to sense temperature, right?
Oh, really?
Oh yeah, they're excellent at it. In fact, they're like this, like the Hulk is like this exposed nerve, right?
Yeah.
That's what a yeast cell is. I mean, they're not homeotherms like us. They don't have kind of normal homeostasis where they can control their own temperature. They're a single cell.
Oh.
Right? And so they're just out there. Whatever happens, right? If the temperature changes, they are at the mercy of that thing. They can't run away. They have no legs, right? So they have to be able to, seriously. So they have to be able to respond in some way. So how do they actually sense the temperature in the first place? That's one trick. And what is the connection to these clumps that form in the cells, which have long been thought to be trash, just some sort of toxic mess, like some intracellular catastrophe that breaks out when you heat up cells. And there the idea was like, you remember this super elaborate protein folding problem?
Yeah.
Right, that we talked about? Well, those are delicate little things. And if you raise the temperature, you sort of, you bake them apart. Okay? And because their structure is very, very delicate, or they're what we call marginally stable. Okay. and they have insides.
That's their molecular structure that we're talking about then?
Yeah, exactly. The chain itself of amino acids, you know, it's fine. It's not going to be compromised by anything. Those bonds are extremely strong, but all the other interactions that are happening don't involve direct molecular, what we call covalent connections, if you remember such things from chemistry. Instead, they're just, they're no worries. I have to say, like, you know, as a brief aside, I am a professor of biochemistry who's never taken a course in biochemistry.
That's incredible.
In part because of those strange career paths, right?
Mm-hmm.
Like it is very nerve-wracking at times to realize that I'm well behind my peers on a lot of the subjects that it is considered classically necessary to know, and yet, you know, I think the fact that we've been able to succeed also reflects that at some level that textbook level expertise is not how you come up with new discoveries, right? You have to be aware of these things, you have to be conversant, but knowing all the nitty gritty is not actually where oftentimes the innovations come from. So anyway, this procedure of like shaking these proteins apart with temperature, you can think of temperature as basically like molecular shaking at that sort of level, then leads to exposure of a bunch of sticky stuff that's normally buried away inside the proteins, now exposed. They're like jelly donuts, right? They're easy to hold on the outside, but they're miserable on the inside. And so then you start tearing these things apart, and they all stick together in some giant mass, right? And that is, first of all, it's useless. Second of all, it's potentially toxic. You have a bunch of, you know, these Death Stars floating around inside the cell. And so you have to deploy some machinery to go clean these things up. And how does that cleanup machinery work? Okay, so that's the broad scale thing of what we're working on. And what I'll tell you is I think that our work over time now has shown that this fundamental picture of what cells are doing, just you can look at things in an entirely different way and see all the same type of phenomena, but they have a completely different interpretation. Okay. Which is that you know, the cell is not surprised by these changes in temperature. Yeast have been around for a really, really, really long time. And so the idea that they're like, oh my gosh, catastrophe, like everything, all our molecules are falling apart. Who could have foreseen this? Like, of course they can foresee it, right? Because they've been through generations and generations and generations, right? So maybe they're not startled by this. What else could be going on? Well, how do you make some biological process extremely temperature sensitive, right? Like, what is the mechanism by which you do that? It's hard to do, right? It's hard to think about, but you have probably been through some procedure where you can see an extremely temperature sensitive molecular process. For example, in your freezer, if your freezer's off by 1 degree, that's the difference between having water and ice. Okay. Cool. That's a phase transition, exquisitely temperature sensitive, right? But these phase transitions, you know, exactly the same as, you know, as boiling. You reach 100 degrees, and that's this narrow boundary between having a liquid and having a gas. Right? So can you have phase transitions inside the cell?
Wait, it's 100 degrees? Did we decide that before or after? Like, did we decide boiling point is 100, or did we notice boiling point is 100 after?
Oh, we just, yeah, in fact, we just define, humans, there's no God-given 100, right? Like,
of course.
On the Kelvin scale, right? I guess I should be able to say this, but it's, you know, it's above. Oh gosh, what is boiling on the Kelvin scale? Anyway, like, room temperature is 370 something on the Kelvin scale, right? So absolute zero is an absolute zero on that scale, and that zero is the only thing that is sort of God-given, right? Like, there's no temperature conceivable below that. So 100 just, you know, that's 0 is freezing on the Celsius scale, and 100 is boiling, and it was defined that way.
Nice. Fine. Yeah, smart. Yeah.
Yes. Yeah. Wise. Not
useful.
Like Fahrenheit. Yeah. Right. 212 and 32. Right.
Yeah.
I don't know. Anyway, so you can see that there's this way for molecules themselves, without any sort of intelligence or cellular, you know, machinery around them, to be exquisitely sensitive to these small changes in temperature. So what if you just had something inside the cell that was doing that, right? Some special molecule that could undergo a phase transition in response to a change in temperature. Okay. Now, it turns out there is a revolution that's happening in biology right now, where, you know, back in 2009, Cliff Brangwyn, now at Princeton, and Tony Hyman, who's the Max Planck Institute in Germany, made this observation that there were whole, what are called organelles in the cell that seemed to behave themselves like they didn't have a membrane boundary. They were in fact themselves coherent liquids, meaning that they had sort of, instead of the usual bit of like a well-mixed reactor that you might think of inside a cell with all the molecules just sort of flowing around in some giant bag, they were able to compartmentalize in a kind of oil and water type of way. There was a separate phase, right, that existed inside the cell. And this observation and the ideas that kind of sprouted out of it have now really spread through and inspired a huge number of us in our individual sort of biological questions. So this, we study the idea that that there are proteins in the cell that undergo a phase transition, that phase separate into these structures in response to temperature. And that is the primary way that yeast cells and other cells may sense temperature. So that's, idea number one is that you can use a phase transition this way. But idea number two is that what would that produce? It would mean that you'd have proteins that were floating around, right, in some sort of,
boundless
fluid bag, right, exactly. And then suddenly the temperature would change and they would phase transition, right? They would clump together.
Cool.
But that's what we observe, right? We observe the formation of all these aggregates. We just interpreted that this was some sort of catastrophe that was causing the proteins to unfold. And so one of the things I like to tell my scientific audiences, and I can tell you, is that despite the fact that this is the textbook idea and you can find it in any number of scientific reviews, no actual protein has ever been demonstrated to misfold in a cell in response to a heat shock. That's an interpretation, not a result.
Right.
It's an attempt to explain what is happening.
Yeah.
And it turns out there's this alternative. And the alternative is that the cell means to do this. It has evolved molecules that specifically do this. It is intrinsic potentially to the sensation of temperature. And that now all this emergency equipment that gets deployed to prevent and undo these things is in fact just a regulatory apparatus for pushing them around in exactly the right way and like causing these changes in cellular organization to be more organized. And that the proteins themselves are not trash, right? When they go into these, these.
So it's a higher order of order rather than chaos, is how you.
That's right. That's exactly right. It's a whole new layer of biology.
That is pretty awesome. Yeah,
it's been very fun. From something as simple as just saying, well, how does a yeast cell sense temperature? You can suddenly get to this kind of, well, at least participation in the revolution. Right? Like
and this is kind of related to a question that I had earlier, but the question I guess I posted before when we talked, and that was about cells that are without a membrane, right? Like and how that might work. And yeah, like obviously I'm curious for different reasons, right? I really like the idea of you think it's like it doesn't matter so much where is life and where isn't life, but for me, like the boundaries and the definitions, that is just super exciting, and it seems like we're getting to a point where there is less of that, right, to some degree. Well, like less definitions, less boundaries. I know your thing is the definitions and boundaries.
No, it's the definitions, like they matter. I think it's possible to get lost in them in ways that are not productive. But of course, it's reasonable to start asking questions like, what is really required, right? So one thing you might think is that a cell membrane is required to have a cell, and that must be that.
That was like the original assumption, right, or to some degree.
To some degree, right? I mean, it's actually origin of life is a big subject, and there's all sorts of ways to get self-sustaining chemical reactions without being compartmentalized. But then there's a second step in which you have to sort of create the idea of a cell that has an identity, and membrane boundaries have long been the kind of way, or sort of a micelle, or one of these small little membrane-bound compartments, right? There's the way that you're supposed to do that, and possibly the only way, because that's what they're now made of. And so how could you ever do away with this? And yes, you're exactly right. What this idea of phase separation does is gives you another way to create these compartments without all the business about membranes, right? You can have just molecules that have this tendency to clump together and do so in a fluid phase, right? This is actually the thing that has lit up a lot of people, is that
seems quite lively to me, right?
Seems quite lively, right?
Yeah.
Seems very dynamic. Personally, I think it's an exploding field, and there's a lot of trendiness and hype around it, and people have fallen in love with the liquid state in ways that I think go beyond the scientific reasons to be excited about it. Right?
Really?
It's yeah, absolutely.
Liquid's super. Water's so exciting to me. Yeah.
Yeah, exactly. I mean, liquid is just great.
Yeah.
And, so there's for a long time there's been this like liquid good, solid bad, and that's just not what we see inside yeast cells. Most of these structures are solid as a rock, and the more you think about it, you're like, it doesn't really matter what that material
That
state
means
is.
that means they're like sort of immovable to some degree. Like the molecular structure is just very tight.
That's right. They're very they're solid. Although you can think about it this way, right? There's a few things going on when you start thinking about what it means to be a solid. Of course, everything is bathed in water inside the cell, and you can imagine something like like monkey bars. That would be
Monkey
inside the
bars?
cell. Monkey bars, or a jungle gym, right? It's like a climbing frame.
Oh, okay. Yeah,
Right,
I know.
from a children's playground, right? Where the thing is solid as a rock. However, you can get into it and get out of it just fine, right? That is, it's a fully accessible structure. It doesn't require any dynamics to have that happen. This is what we call the idea of mesh size, right? So you can have a solid structure, but you can also have one that is very solid that you cannot get inside, and that nothing can penetrate,
I
right?
see.
So chemical reactions would be impossible, right? Because you can't get the substrates in, you can't get the products out. But you can have these other structures that allow that sort of exchange. And people have made this kind of, it's
Like our skeleton
cool.
to some degree, I imagine. Well,
Yeah,
it's not
exactly,
so solid.
Right? Right, all sorts of stuff can go, go in and out and so on. But yeah, the idea that like the activity that you might be able to encapsulate in one of these structures is not dependent on it being fluid. It's just dependent on it having a mesh size, right? That allows certain things in and certain things coming out. And it's those sorts of, it may seem like sort of an intellectual distinction, but like thinking about the actual biological problem that's being solved and thinking about how that biological problem maps onto a particular material state, right? That's the
Because like
question.
solids, I guess solids have like different boiling points and stuff like that too, right? So it must make for more resilient structures in some ways. And
Well, I mean, yeah. So in most cases, you're going to get, some interaction between the strength of the interactions and their solidity. But there are other ways to get solidity. I mean, it's a really fun subject, right? It's like, yeah, I hate to give you the it depends. One way to think about liquids is that you can have facilitated liquids. That is, imagine a bunch of people in a room and they're all just holding hands or grabbing each other's sweaters or whatever, right? And, you say, everybody hold on. But then you have somebody come through and say, okay, you let go for just a second, you grab this person, right? And you have somebody rearrange, right? Another kind of molecule that can come in and stir the pot.
cool.
From a distance, that thing might look like a liquid, even though locally everything might be absolutely fixed.
That is cool.
Quite exciting to think about the sorts of, what we like to say is what's the functional repertoire of these things when you can get a bunch of molecules together or spread them apart, right? And that's typically not how biologists have been thinking. We think about, or biochemists at least, right? You think about, okay, here's an enzyme, it does a reaction. What reactions could it do? An individual molecule. But now you're talking about connections between molecules.
That is so cool.
Yeah, how they talk to one another.
So it's like a higher level of complexity. It's a level up and trying to understand the communications and.
How do you build your army?
That is so cool. That is awesome. Yeah. This is a lot to think about for me. I really enjoy this type of conversation because it provides so much perspective for me. And
Likewise, by the way, you're a
I'm just
host.
inquiring. I'm just inquiring in that.
You do a very nice job of this. So
thank you.
I'm very happy to sort of come along.
That's awesome. And this is then the most recent project that you've been working on?
Yeah, it's been sort of the build. My graduate work I said was all theory, and it was thinking about this relationship between errors, maybe not errors, right? Changes that occur in the translation of those messages into proteins, right? So the mistakes or what have you, right? That the ribosomes make when they're actually taking that message and doing that last translation. It turns out, I told you, you can make 1,000 E. coli without making your first error, right? And so the copying machinery is usually incredibly precise, but ribosomes are terrible. They're making tens of thousands of proteins, but the average protein, right? You make 5 proteins and you make 1 error. So
Wow.
their error rates are astonishingly high. And you're like, and that's the place where it seems to really matter, right? Like it's the last moment where the genetic information is being expressed into something that's going to have a function. So wouldn't you like micromanage that last step? The answer is no, right? Like they don't at all. How can they get away with that, right? So my graduate work was basically saying, look, you can build proteins so that they are robust to these mistakes. So they can still work despite having all sorts of these mistakes in them. And that is thinking about the sequence space that's accessible to those proteins and how that maps onto also these processes are being disrupted by mutations, right? A mutation might go in and cause the same kinds of bad effects that heat does, causing the protein to misfold, lose its structure, lose its function, and cause a catastrophe in the cell. And so coming from that world, that's what sort of established my kind of scientific home. You can see it's closely related to the sorts of stuff that Frances was doing, but different enough so that I could have an independent life, which she really helped me, of course, with. And then building that into, okay, well, what are the proteins that actually misfold in cells, and what are the effects of errors? And that led us to simple questions about, well, what happens if you do to cells what everybody does to make proteins misfold, which is heat shock them. And, oh, this doesn't look at all like what we're supposed to be seeing, and that's interesting. And you can start making empirical discoveries. So this is now the whole lab is working on different aspects of this problem of how you sense and respond. What's the original? What molecules are the most sensitive in the cell, and how does the cell change its mind? What is it really doing when it goes through this process? It's like the heat shock tells it information about the world.
Yeah.
It tells it that the temperature has changed. The fun thing is actually that like these yeast cells, like where do they experience these sudden temperature changes from like we're happily growing at 30 degrees Celsius or 25 degrees Celsius, and we suddenly experience a 42 degrees Celsius change. Where does that happen? Well, you've just won the lottery, yeast cell. You've just been eaten by a bird. Birds are 42 degrees Celsius on the inside.
That's okay? Cool.
Right? And the yeast, like we've established before, right, have no legs. And so how do the yeast get around the planet? Well, they go through a bird, for example. They're dependent on animals for dispersal across the planet. So they're delighted. But all they have to do is take this new information, which is we have left the grape that we were living on, which was a really wonderful home, right? We're now in this very inhospitable environment that's hot, and we need to prepare to be pooped out somewhere, an arbitrary location
that's so cool.
on the planet, right? So they have to change their minds. Like we were working on this one thing, it was great. It was a party. Now buckle down, people. We were like, we're going to go into spore mode. We're going to go, we have to change our minds.
And that's the circle of life, right? In essence. And
that's right. I mean, it's this evolutionary ecological thinking coupled with molecular thinking. Toward nature is kind of like, I don't know if it's fair to say guiding these processes, but they all kind of seem to fit somewhere. Yeah.
Right.
Nature is these processes. Right?
Yeah, exactly. Oh, that's so cool. And yeah, I was gonna ask, as far as like having a lab is, because you hear a lot about like when you are doing scientific inquiry and how that resolves into practicality. Like, is that something that you are much bothered with? And I like that you brought up Frances, because yeah, when I was thinking about like how do you make practical use of this, it what I thought back to was the example of the laundry detergent, right? Is that fair, or like is there practical application for some of your understandings yet?
Yes. I mean, it's not my focus. It's not what gets me up in the morning.
No, but it's like if you get funded or like these people have to be interested in you producing something.
It's not hard to think of ways in which you can make this sort of information practical if you can, right? Like, so why in the world? I mean, I think all fundamental scientists, right, of my sort of ilk have to challenge themselves. For like, what is the reason why taxpayers, right, or nations should fund this work? Because I'm funded by you.
Well, I'm just curious, but there's someone that's gotta be.
That's right. So I do, I take this very seriously, and so let me give you a few examples. One not from our lab, but I think it's just a very, very good example. Budding yeast. Why would you study budding yeast, right? Fine, you can make beer and bread out of it. Fine, maybe it's our ancient microbial ally, but it's not a human, right? Why would you study humans and how human cells work? And so we can fix humans and do good things for humans. Well, first of all, you can study something that has a very similar cellular structure to humans, right? Which yeast cells do, because we're both eukaryotes, it turns out. We have a nucleus, for example. That's one of the things that makes a eukaryote. And we keep our DNA inside that things. And I can study that yeast cell much more cheaply, right, and much more ethically than having to go off and, like, say, collect a bunch of undergraduates and grind them up for material, right? Centrifuge, right? That's not allowed, right? for obvious reasons. So I can maybe move farther and faster, right, by using this organism. And so if that's true, then you should be able to find examples where this has actually worked out. So, you know, one great example is Randy Sheckman, who studied protein secretion in Saccharomyces cerevisiae. Okay, this is idea that there are many membrane compartments inside cells, and you have to move proteins from one side of the membrane to the other side of the membrane, okay, across multiple membranes, eventually outside of the cell. So, you know, studied that process, and then realized that it could be manipulated, and understood all the molecular details, like worked this stuff out, you know, Sheckman and many other people. He won the Nobel Prize for that work, but, like, who cares? Well, 1/3 of the insulin, right, that diabetics inject is now produced in yeast cells.
Oh, cool.
Right, and it's secreted through the same pathway, right, that was discovered, right, and studied in detail. And we could never have done that, right, without having studied the, you know, these yeast cells are useful as little biological reactors.
Cool.
You know, another example is, you know, that.
And we have a lot in common with fungi too, right? It seems like, as far as yeah, that's cool.
There's this, you know, there's people in my line of work often talk about, you know, this is evolutionarily, this feature or molecule or whatever is evolutionarily conserved from yeast to humans, as if this is some, like, you know,
like we used to be mushrooms, huh?
Well, you know, they mean on the tree, right? Like, you know, you can imagine the entire tree of life, and, you know, here's us here, and here's yeast over here. But the reality is, it's not that way at all. It's like, here's yeast, and here's humans, and then, you know, here's, like, plants, and then here's, you know, pathogens, and, you know, giardia and protists and all this sort of, you know, the tree of life is vast, and we're actually right next to yeast at some scale. We're actually quite similar to them. Of course, we are quite different too. They're single celled, we're multi-celled, you know, all these differences. But yeah, so the other bit is, like, you know, thinking about temperature sensing. Well, we discovered, because we were looking, you know, for what are the actual changes that happen in cells when you heat shock them. We found these proteins that would clump together. We then identified the molecular domains, right, the parts of the molecule that are responsible for temperature sensing, and realized that you could then take those bits of protein and stick them onto other proteins. Actually, genetically, you just you add the little bit of DNA that encodes that thing to the gene, and it makes a protein that's slightly longer and has that little thing. And suddenly, that new protein would clump up in response to temperature. Well, now you can control matter, right? You can control matter in response to temperature, right? We have this transportable domain that allows you to do this. And so we created a method of protein purification, a way to make a lot of pure protein, like we were talking about making subtolysine, or to put in the laundry detergent, right? Where you can use a clever mix of temperature perturbations, right? Change the temperature, and then use centrifugation, which is very cheap, right? And just spin something down, and so, you know, big assemblies go to the bottom of the tube, and the small floaty stuff stays up at the top. And just those two operations, changing temperature and using a centrifuge, allows you to get pure protein with no sort of special apparatus. And we do this with high school kids, right?
That's so cool.
It's an unexpected, right, serendipitous discovery, and I think science is filled with these sort of things where you don't. Yeah. Sometimes the solutions that you find are not the ones that you're originally looking for.
Yeah, absolutely. Yeah, I guess I was wondering, just in terms of like, if you get any pressure as a scientist to do anything other than just, you know.
I mean, people will ask.
Explore, yeah.
But and yeah, it's not just exploring. I think there is a.
Like a general direction, maybe, that you have to explore.
No, yeah. So many people get into science because they don't want to be told what to do.
Yeah.
That was not my case. Actually, I loved working I had a very strong set of bosses in industry, and I loved working for them. And Francis was an outrageously great boss, right? The thing that you want from your boss, it turns out, is, like, not to not have one, but to have one who's really great and who can bring out your best and inspire you and take you in new directions that you wouldn't have dreamed of, right? And I think that's the sort of leadership that I have now tried to do with my own people, is be a good leader.
Awesome.
But nobody's hovering over me and saying you should do this and you shouldn't do that.
That's cool.
There is a side pressure of, like, we write grants. If those grants do not get funded, right, and somebody decides that this is not practical or not useful, or for whatever reason, then you don't have money to do that work, and then your lab shrinks and you go out of business. Now, what I have found is that I can honestly talk about what I'm doing, and I can say it in terms of yeast cells, and I can say it in sort of basic terms that I think are true, and I don't try to smuggle in a bunch of stuff about disease or curing, things like this, and my grants still get funded.
That's awesome.
Because people recognize you need a portfolio of bets as a nation, right?
Yeah.
And some of those bets are not going to pay off for 20 years, but when they do pay off, they're going to have a dramatic effect on our understanding. So that's the standard that I like to hold myself to and my lab to, is how are we really changing how people think about some vast swath, right? Where that's your impact, right? If you're just going to discover some molecule that does something, yeah, it's useful. It is a discovery, but for us, it's not you have to rewrite the chapter or rewrite the textbook.
Yeah, that's great. I think I love that because it's something that's concerning to me about the way that I perceive science oftentimes, where it needs to be practical sometimes is how I see it from the outside. Like maybe there's not enough investments into, just exploring the universe inwards and outwards. And I love that, you're getting to do that.
Yeah,
so
and there's,
that's awesome.
I think it's important to vote for people who you think have the perspective that you would like. I mean, this is how it happens, right? And I think there are these modes where people get very frustrated that, if people are suffering, right, we should not be spending our money on like looking at how fruit flies have sex. Like, well, actually, let's talk about what we have learned from studying how fruit flies have sex, right? And and let's talk about what we've learned from studying how yeast cells secrete proteins, right? You can do these shockingly great things that we've never would have discovered. But I think that's an honest conversation that people have to have at many levels, right? To sort of get the right mix and get the right people working on the problems that.
Yeah, I mean, it's not like human curiosity is not any less important than any other human activity from my perspective, right? It's just like it's something that we crave, I think, understandings and just.
That's right. For the cosmologists, right? What's at stake if we do or do not understand the origin of our universe? Well, at some level, the question itself has value, right? It tells us where we came from. It maybe tells us where we're going. It maybe tells us ways that we could change the fate of things at much longer timescales. I'm with you. I think, you give people exposure to the world, and you sift out those who have these kind of questing minds, who have a curiosity that's not completely untethered, but maybe quite untethered to the traditional ways of thinking. And don't give them a ton of money, but give them some, right? And if they succeed, then you give them a little more, right?
Yeah.
There's a process. Like we're not just like suddenly we win the lottery, right? Like I had to come up. I'm now 18 years into this journey, and I had to earn my way along the way.
That's incredible. Well, Allan, I really hope that we stay in touch and that we'll talk about stuff like this at some point else. Maybe I still have so much unconscious and subconscious processing to do from your conversation, because a lot of it goes past me and I get like a gist and I'm like, let me pin that down because I think I got something from that. And it's just so wonderful. But I'm sure in my sleep I'll process more of it and I'll come back at you with more questions because it is just so fascinating to me.
It will be my absolute pleasure. Thank you so much for having me. And absolutely, I still cannot believe that I'm talking to you. Like,
likewise, man.
Heroes, right? So,
please.
So thanks for this, and look forward to the next time.
Yeah, and here is your Twitter. Everyone should follow him. It's Allan Drummond. Drummond. I am gonna mess that up every time though. And of course, Instagram. I think it's the same, or is it the same name that you use?
Slightly different. D.A. Drummond on Instagram and D. Allan Drummond on Twitter.
Yeah, for more information about your lab and on this website, there's a lot of papers and other research. Here's where I read a little bit about Francis as well. So you guys should all check that out. And if you have any other shout outs?
Just a shout out to my lab who does the actual research. I mean,
these guys.
I've not done them too much violence, and also to my historical mentors who got me here, particularly Francis, we heard a lot about today. I'm terrified by the idea that you might watch this and sort of go, "That's not what I said."
I'm sorry, Francis. I'll take full responsibility.
You should look up these people. It's really fun to be a scientist, and if anybody's out there is interested in kind of like seeing how to be, what it is that we do. Give me a poke on social media, and I'd be happy to engage.
Amazing. Well, like I said, we'll keep in touch for sure. Thank you very much for joining us. And yeah, we'll see you next time.